I'd like to thank first the Van Andel Institute, Dr. Trent, and Dr. Scholler for inviting me here today. I'd also like to thank Dr. Scholler for being such a great collaborator and a friend, and most importantly, an inspiring human being. I really like to say that, and I mean that. You just stole my introduction basically on what I was going to say about how this trial came to be, but that's great. Thank you very much. This is the place where I have done all the research. This is the new cancer center that we are currently building. It's an NCI-designated cancer center in Honolulu. And as Dr. Scholler said, I was recruited to the College of Pharmacy recently to head the Department of Pharmaceutical Sciences, a department with about 16 tenure track or tenured professors. And so we continue. I'm still a full member at the Cancer Center, and we continue to work together. Well, this story for me began in 2001 when I became interested in neuroblastoma and became an assistant professor in the Cancer Center, and then went to my first neuroblastoma conference in Paris, Advances in Neuroblastoma Research, ANR. I was at the time very familiar with an enzyme called ornithine decarboxylase, or ODC, abbreviated, and I did not know, quite honestly, very much about neuroblastoma at that time. I knew it as a cell culture system, but not really ever had any contact with parents, so I went to this conference, and I realized that NMIC amplification is extremely important, as you all know, in neuroblastoma. And I also knew that NMIC, as much as c-MYC, is a transcription factor that activates ornithine decarboxylase. So my next thought was, well, I mean, has anybody in the neuroblastoma community ever considered to treat neuroblastoma with an ODC inhibitor instead of maybe looking for NMIC inhibitors? And I was sure somebody did, and I went back to my hotel room, and I found very little literature, surprisingly, that would talk about this. I mean, there were papers in the 80s, but it was not really clinically oriented. So I decided to write a little grant and was funded to basically look at ODC in neuroblastoma and to look whether this drug DFMO would be effective. Now, DFMO is an old drug and had been used in cancer trials long before I thought of it. So I only connected the NMIC, ODC, and neuroblastoma and thought that maybe DFMO could be a good drug. DFMO had been used in monotherapy cancer trials in the past and was in the way that trials were set up, not extremely successful at the time, but I thought neuroblastoma is a different type of cancer, and I think it's worth a try, especially because the drug is very soluble, drinkable, and also has very low cytotoxicity. So what happens with ODC, let me switch to the next slide to explain this. Okay, well, I would like to say first that, so I basically walked away from this conference and felt that ODC is a novel, quote-unquote, therapeutic target for MYC-driven neuroblastoma. And I also, for the first time, met with parents in the neuroblastoma community, which was a very, very important and significant moment for me, because I really realized that neuroblastoma is not just a cell culture model, but it is, and I'm a basic scientist, so, but a severe disease that affects many, many children and parents. And so from that moment on, I looked at this as a completely different project, or as a passion to really see if we can find something and put the patient up front and see if we can find something quickly and not just discover brand new drugs, but maybe repurpose an old drug like DFMO. Now, we just happened to hear about Nifurtimox and Chagas disease, and interestingly, DFMO is also used by the WHO to treat something similar to Trypanosoma disease called African sleeping sickness. And after melarsoprol, DFMO has now been recognized as the primary treatment, combined with Nifurtimox, which is somewhat interesting, coincidentally. So this is now, and it's called the NECT treatment. Nifurtimox, Eflornithine, which is another word for DFMO, combination treatment. And so DFMO, as I said, is an old drug, synthesized in 1978. The molecule is right here. And was used by Sano Aventis, actually given for free to the WHO for 5 years to treat patients in Africa. They use the IV formulation, not a oral formulation as we are using here in the neuroblastoma trial. So what happens, most people don't know what these substances are. They're called polyamines, putrescine, spermidine, and spermine. And the bottom line is, without going into too much biochemistry detail here, this enzyme, ODC, converts ornithine to putrescine, and then further to spermidine. Spermidine. These substances are highly upregulated in proliferating tumor cells. And so the therapeutic goal will be to reduce those compounds in the cancer cells in order to stop the tumor cells from proliferating. Now, MICN, as I said earlier, activates the ODC gene, and as a consequence, ODC enzyme is upregulated, and these substances are produced. And it leads to proliferation. Now, if we inhibit this with ODC, which is a suicide inhibitor, an enzyme-activated irreversible inhibitor, we reduce those polyamine concentrations in the tumor cells, and as a consequence, the cells are arrested in their cell cycle. Now, I don't need to introduce neuroblastoma to this audience. Obviously, all I just wanted to point out again that for us, in particular, the NMIC amplification was the main reason why this all came to be. And so this is an image that Dr. Scholler provided and actually led to this, I wanted to say here, that's her image that we were able to get a cover in the International Journal of Cancer when we published a paper on ODC in neuroblastoma. So what happened first? I mean, as a basic scientist, I went back to my lab and we started to look at in vitro in neuroblastoma cell lines how DFMO works. And we found that after about 3 days of treatment, these cells were dose-dependently inhibited. These are all neuroblastoma cells in a dose-dependent manner. And so that was encouraging. And we then, here's just a morphological view of those cells. You can see here control cells grown after 3 days with DFMO treated, sort of a differentiation, but we call it elongation. It's not really differentiated like it would be with retinoic acid, as Dr. Thiel was talking about today. And here, NMB7, both are NMIC amplified cell lines. And if you treat with DFMO and add in one of those polyamines that I just mentioned, you can rescue those cells pretty much to about 80% again. And so that shows that really the effect that you get is due to the depletion of these polyamines, and spermidine in this particular case. Now, this is a bit busy. I just wanted to summarize. We then went on to do research to look at where exactly in the cell cycle is the impact of DFMO, and we found that the G1 phase is arrested, and we did flow cytometry to do these experiments. And we looked at much more carefully on a molecular level which proteins and cell signaling molecules are being upregulated and modulated by STS page and Western analysis. And I'm not going to go through this now in detail because I really do not have the time to do that. But bottom line is that we found that P27, which is a cell cycle dependent kinase inhibitor, was upregulated, and RB, the retinoblastoma protein, was hypophosphorylated, which is one common pathway that occurs to be activated in G1 arrest. What was really interesting was that NMIC was downregulated on the protein level too in response to DFMO, and heavily downregulated. As you can see, after 3 days here, it was almost non-existent. These data were then published first in 2005 in Oncogene and in Cancer Research later, a follow-up study where we looked at more phosphorylation. These are my postdocs and students in the lab. We also did a study with Dr. Gertz in Amsterdam, a neuroblastoma lab in Roger Versteeg's lab, where they have data sets, tumor samples from about 110, or in this case 88 tumor samples from patients, and we looked at what effect ODC expression had in those samples against overall survival probability. And as you can see, low amounts of ODC were present in those tumor samples that had a very high overall survival probability over several years. These are months. Sorry, I got cut off here. These are months. And if ODC was highly upregulated, the overall survival probability drastically decreased, and it had a very high p-value here. So this was published in this particular journal that I had pointed out earlier. So then came the AACR in 2008, met Dr. Scholler at that conference, and as she already said, I was introduced to Scott Kennedy and Neil Hodgkinson, who were the two gentlemen that I talked to, and they mentioned that I should talk to Dr. Scholler about this. And so it really was the parents that brought us together, as Giselle said. So we did, after a few phone calls at the conference, sit together, and we started talking about this project, and then came up with, we tested a few different combinations again, and these are now in vitro data from Dr. Scholler's lab that we presented more recently at the ANR in 2010, showing that DFMO combined with etoposide has synergistic effects in these 3 different neuroblastoma cell lines. Dr. Scholler then went on to look at in xenografted mice with neuroblastoma tumors to see the effect of these combinations, and as you can see in the upper panel here, you have the tumor masses showing here in those mice, and if they're treated with DFMO and etoposide, the tumor mass is in some cases still present, but significantly decreased, as also is shown here quantitatively on this figure.